information that you're interested in. That's a good way to think about introns. They're nothing the ribosome is interested in because they're not encoding any amino acids. Are they necessary? Apparently not, because prokaryotic genes do not have any introns. If you look at a prokaryotic gene, it's only exons. It's only protein encoding codons. So obviously you can have genes without them, but we eukaryotes have them and prokaryotes don't have them. And maybe one more quick point. In this diagram I'm showing you right here, I'm showing the gene as having 3 exons and 2 introns. That's not, I don't mean to be saying that that's always the case. Some genes might have 7 exons, and if you think about it, 6 introns, because the introns are in between the exons, so you'll always have one less. And some genes might have 10 exons and 9 introns, spacing them apart.
They believe that that's done because there's a need for more diversity in eukaryotes?
Yes, the shorter answer is nobody knows exactly why they're there, why the eukaryotic genes are interrupted by these introns, but they have a couple of ideas, and one of them does relate to what I guess what you call diversity, that they think that the gene is modularized, and that helps you get more protein from just one gene, and I'll expound on that idea in a little while. But yeah, they think it eventually it has to do with eukaryotes being more complex than prokaryotes. Okay, now getting back to my mountain magazine analogy, if you think about the exons as being the parts of the magazine article that you're interested in, and the introns being like a bunch of useless advertising interspersed with your magazine article that you're not really interested in, everything else being equal, you don't want to see the advertising. It's just going to make things more confusing for you. And if you're a ribosome, you really don't want to see the introns because they're going to really make things confusing for you. If you were a ribosome and you're translating this into amino acids, you'd say, okay, AUG, I make methionine, so you'd start off the protein correctly. You'd say, okay, I'll make a methionine, then the next codon is CGU, and so you would make a arginine, so that would be correct for amino acid number 2. Then amino acid number 3 is encoded right here, AAG, and so you would make a lysine, which is correct. Okay, there we go. But what would your 4th amino acid be? Or maybe I already told you guys that. If you were the ribosome and you actually read this intron, what would your 4th amino acid be? It would be GUA, which is valine. And that would be a mistake because valine's not supposed to be part of the gene that comes from this protein. And your next amino acid would be incorrect also. You would read, if you were the ribosome, you would read an AGU, which is serine, which I guess that comes later on, but the point is it doesn't come next, it doesn't come yet. And so these introns would totally and utterly make the wrong protein if they were allowed to stay in the mRNA. The ribosome would make the wrong protein, and so they have to be cut out. And that was the analogy I was trying to make. You would like it if somebody would cut out all the annoying advertising from your magazine article, and so that's what's going to happen before the pre-mRNA is allowed to leave the nucleus. All these introns have to be snipped out of the pre-mRNA. And so that is the 3rd and probably the most important part of the processing of the pre-mRNA before it leaves the nucleus. All right, so it's called RNA splicing, cutting out the introns from the mRNA and joining the exons together before the mRNA can leave the nucleus. How is this done? Well, there are sequences that mark the beginning of an intron and the ending of an intron. In other words, as far as the cell is concerned, it can read the sequences around the exon-intron borders, the exon-intron junctions, And so the cell is able to tell where an exon ends and where an intron begins by those sequences you find at the borders. And so by keying in on those sequences, there are enzymes in the nucleus that are going to be able to splice out the introns. And in this diagram, I'm going to make the pink the introns and the yellow the exons. And so what these enzymes do is essentially this. They bring the exons closer and closer together, and the introns therefore kind of loop downward like that. And eventually these enzymes entirely splice out the introns, that is, just completely cut them out of the pre-mRNA, and ligate, which remember means join together. They ligate the ribose phosphate backbone of the exons to join all the exons together into one complete mRNA transcript. And what happens to the intron, G's and A's and U's and C's? They just drift off, and eventually in the nucleus those are going to be torn apart to their nucleotides again, and I guess recycled into new mRNAs. Remember I mentioned a moment ago that the average gene from the start codon to the stop codon is around 8,000 base pairs in length, but the messenger RNAs that are made from those are not 8,000 base pairs, because when you cut out the introns, they're going to get smaller. And I guess I don't know exactly what the average is for a messenger RNA, but just taking an estimation, probably somewhere around 1.5 kilobases. So there's it's quite a bit shorter. It's probably about only a fourth or a fifth the size of the original pre-mRNA by the time you cut out the introns. And so now it is the mature mRNA, and in that when the 5' cap and the poly A tail are added, then it's ready to exit the nucleus. Let's look a little bit more closely at the actual splicing of the pre-mRNA. So again, in gold we have the exons that actually encode the amino acids, and in pink we have the intron G's and A's and U's and C's, which have to be cut out so they don't confuse the ribosome. And as I said, there are sequences right at the border between the two that can be recognized by enzymes. All right, there are some enzymes in the nucleus called small nuclear ribonucleoproteins. There's a mouthful. And they're just called SNRPs for short. That's the only thing I really call them by. And it's a good name because it sounds like SNP, which is sort of what they're doing. Anyway, these small nuclear ribonucleoproteins, these SNRPs, recognize the sequences at the exon-intron boundaries and bind onto them. They have binding sites for those particular sequences. And so some of them cluster here at the beginning of the intron, and some of them cluster here at the end of the intron where it borders with the next following exon like that. And then the SNRPs that have clustered here at the beginning of the intron and the SNRPs that have clustered there at the end of the intron start coming together, and that loops the intron downward. Let's see, like this. All right, so there in pink you see the intron's been looped downward, and the two exons, the more upstream exon and the more downstream exon, have now come together. All those SNRPs that are now clustered together are now called a spliceosome. All the SNRPs, and some other enzymes get involved also, and they all cluster together, and they're now a spliceosome. And the spliceosome is going to do several things. The spliceosome is going to physically cut off the introns, just basically snip them free so they're no longer part of the mRNA. And the spliceosome is also going to enzymatically join the backbone together so that the more upstream exon here is physically joined to the more downstream exon there. And so you get one continuous piece of mRNA of just the exons joined together, and now the introns have been spliced out. And there it goes. Sound clear enough? Should strike you as odd. I mean, it strikes me as odd. I've been teaching it for several years, but as long as you understand the basic concept of the events that happen, that's the important thing. All right, so maybe let's take a step back here and look at the pre-mRNA. Remember, this is the one that just rolled right off the gene. Initially, the average pre-mRNA is around 8 kilobases long. It's got that 5' cap that's been added to this end. It's got a poly A tail added to that end. Part of its sequence are the exons, which are the parts that actually are going to be read by the ribosome and turned into amino acids. But part of the pre-mRNA is also these introns, which we need to get rid of before it exits the nucleus, because they would confuse the ribosome with their extra nucleotide sequences. And so it's oh, go back. There it is. So those introns have to be cut out. Now, there's a question Rolando asked earlier about what the function of these introns are. And as I said at the time, nobody really knew when introns were discovered why they were there, because prokaryotes seem to get along fine without them, so they're not absolutely necessary. And so people try to figure out, well, do they do some good? Is there a good positive function for them? Because at first they seem just sort of silly and wasteful of the cell's resources. I mean, why have nucleotides if they're not being used to encode any amino acids? Well, to begin with, some of these ideas about why eukaryotes have introns. They are found in the gene also. I've been emphasizing what goes on with the pre-mRNA, but of course the pre-mRNA is transcribed right from the gene. So if we find introns in the pre-mRNA, it's because there are introns in the genomic sequence for this gene also. So the question really is, why do the genes themselves, the DNA genes, have these introns built into them? And there's two leading theories about why eukaryotes have these introns. And to understand both theories, you have to understand this little blurb of text right here, that the exons are usually what are called functional domains of the protein that's encoded. Let's go back to something, to explain these functional domains, let's go back to something we saw earlier in the semester. When we talked about signal transduction, I mentioned something called tyrosine kinase receptors. These are proteins that are found in cell membranes that bind a signal molecule. Here's the part of the protein that binds a signal molecule out there. Here's the hydrophobic transmembrane domain. And this intracellular part right here has some enzymatic activity. Here's the signal molecule. Now, if you remember way back at the beginning, a few weeks ago, these tyrosine kinase receptors have to dimerize after they bind their signal molecule. But after they do, then the intracellular part starts functioning like an ATP binding enzyme. But what I'm emphasizing here is different parts of this protein are doing different things. The part that's sticking out there outside the cell, its job is to bind the signal molecule, the ligand of the receptor. The part more in the middle of the protein, its job is to anchor the receptor to the cell membrane. In other words, these have to be hydrophobic. This is a hydrophobic domain of the protein. And the part that's sticking out down here of the protein, that has an ATP binding domain. It acts like an enzyme that binds ATPs and then takes one of the phosphates off the ATP to phosphorylate some tyrosine. And so it's really one protein. It's a bunch of amino acids linked together, and yet different domains in it have different functions. This domain is for binding a signal molecule. This domain is for anchoring to the membrane. This domain is for binding ATP and ripping phosphates off ATPs. And so what they found is in most cases, the exons correspond to these different functional domains of the protein. If this is the mRNA that encodes the protein, the first exon, or at least the first group of exons, would encode this domain of the protein. The second exon or second group of exons encode the hydrophobic transmembrane domain, and the third exon, or at least third group of exons, encode the ATP binding domain of the protein.
So then the protein's function is a combination of the coding in the DNA and of the splicing? Because it seems, is it that something like there's a template exon for, let's say, receptor sites.
Right. Okay.
Template exon for transmembrane membrane.
Yes.
And so you can put a different.
Yes, I think that's exactly. I guess I'd say it's the exons allow the genes to be modularized. This exon is an exon for making a protein that binds this signal molecule. This exon encodes a protein domain that allows things to be anchored to a membrane, and this exon encodes a protein segment that allows proteins to bind ATP. And all of those, even in and of themselves, are very useful segments of proteins. And so by modularizing them into different exons, as we'll see here in a moment, allows the cell more flexibility, and it might help evolution in mixing and matching exons together to create new proteins. That's one of the theories that we talk about here. So yeah, that's exactly it. So anyway, think of each exon as being like a little microprotein that encodes like a microprotein, a small segment of a protein with its own particular with its own particular function. Okay, darn it! Just when I was getting into the exciting part, we ran out of time.